Welcome, and thank you for joining us for our live virtual MDA Engage Pompe Disease Symposium. While we would have loved to be in person for this event, we appreciate all of you joining us virtually today. My name is Elise Qualley, and I am the Director of Community Education for the Muscular Dystrophy Association. We are so glad to have you join us today for this important educational symposium. MDA is committed to educational for our community. We believe in the power of bringing our community together for opportunities to learn from specialists and having opportunities to connect with others. This event is part of a larger MDA Engage series with disease-specific symposia, community education seminars, and community webinars as well. Our community education seminars are geared towards general neuromuscular disease topics such as best practices in care, research and clinical trial updates, and genetics. While each event will feature regional specialists covering these mentioned topics, each event will also feature a specialty topic which can be viewed on the screen. To view the agendas for these upcoming seminars and to register, please visit the MDA Engage section on mda.org. We would not be able to put on our educational programs without the support of our generous sponsors. I would like to thank Sanofi Genzyme, Amicus Therapeutics, Spark Therapeutics, AvroBIO, and Diplomat Specialty Infusion Group for supporting this Pompe program. I would also like to thank their representatives who are in attendance today. Following the event, you will receive an email that will provide some additional educational materials from some of these companies. So be on the lookout for the email that will come from MDA Engage at mdausa.org. Now, before we dive into our Pompe Disease Symposium this morning, I do have a few housekeeping items to go over. We are recording most of today's event and we'll be posting it to mda.org website for on-demand viewing in a few weeks. For those of you joining the event live, please know all phone lines have been muted. We will be having a Q&A session at the end of each presentation, so please utilize the Q&A icon to type in your questions. If you hover over the bottom of your screen, a tray of webinar icons will appear. Click on the Q&A icon to open the feature and enter your question. You don't need to wait until a presentation is over before you submit your questions. Any questions that come up during the presentation, please feel free to share those in the Q&A box. We encourage participants to communicate with other participants by utilizing the chat feature throughout the day. Want to just say hi to let participants know you're attending? Want to share a bit of your story with others who are on the line? Or have a question on something outside of the topics we will be discussing today that you'd like to obtain suggestions from the community? Please use that chat feature, which is also found down on the bottom part of your screen. You will notice on the agenda today that there are small breaks between some speakers. Please feel free to stay connected to the live broadcast during these times, but this will allow you time to grab some water or for a break throughout the day. I would like to thank my colleague, Nicole Petrowski, who is our Community Education Specialist, for assisting today, as she is running the technology for this symposium, and she will be assisting in closing out the event. Finally, we will be sending out a brief survey of the event by tomorrow morning, and we would like to receive your feedback on what you heard today. We want to make sure we are discussing topics that are of interest, and we use your feedback as a way to improve future educational events. So thank you in advance for taking 5 to 10 minutes to complete the survey post-event. With that, I would like to say thank you to our next speaker, Dr. Heather Lau from NYU Langone Health. Not only is Dr. Lau speaking today, but she has been instrumental in helping bring this symposium together as serving as our event chair. As a little introduction to Dr. Lau, she is the director of the Lysosomal Storage Disease Program and Associate Director of the Division of Neurogenetics for NYU Langone Health, and Assistant Professor in the Department of Neurology at NYU Grossman School of Medicine. With that, Dr. Lau, I'd like to say welcome and thank you for being here today and for kicking off our symposium, sharing a little bit about what's top of mind for everyone right now, and that is COVID-19.
Hi, good morning. Thank you for having me here today. It's been a pleasure to be part of this conference, going back to March when we were supposed to have this in person, and then we were unfortunately delayed and had to have this now. But with that, I want to talk a little bit about what delayed us, which was the COVID-19 pandemic and the impact on Pompe disease. So I'll just go through this a little bit. Here are my disclosures. I work with pharmaceutical companies to help run clinical trials through phase 1 through phase 4 in both small molecule, enzyme replacement therapy, and now on gene therapy in a variety of lysosomal storage disorders. And it's an integral partnership with pharma to help bring these therapies to FDA approval. So we'll talk a little bit about the multisystemic manifestations of COVID-19 and specifically with its impact, potential impact on Pompe disease and how it's impacted our access to therapy for our patients with Pompe disease, and a little bit about exposure and what to do and some ethical considerations. This talk was generated in real time as I was working with our frontline workers in NYU to understand how this coronavirus, this novel coronavirus, was impacting our patients, looking how it potentially could impact our patients with lysosomal storage disorders. And so looking at today, the CDC data, as of today, we have confirmed cases of over 3 million. We have 131,000 deaths, but we have close to 1 million recovered. And we can see here on the map of the United States where there are more than 100,000 cases. And whereas with New York, we went through our apex of the height of the pandemic in March, April, we are now coming down and finally feeling some reprieve. However, unfortunately, there are other areas of the country that are now just peaking and sadly are going through exactly what we went through in New York. So hopefully I can share some of my experiences with you all so that you can understand what we went through and maybe apply it to what's happening in your state or your community. So as most of us know, the typical symptoms of COVID that we recognize, overwhelming majority have some form of fever, a cough, and they describe fatigue. There could be some productive cough, not just a dry cough, but productive cough. And then shortness of breath, myalgias or muscle aches and pains, sore throat, headache, chills. But there are other manifestations of the disease that we've become aware of, including some non-specific GI complaints, including nausea, vomiting, and diarrhea. And there's also this interesting concept of loss of sense of smell or taste that might even precede any other symptom. And there are a whole bunch of patients that are mildly symptomatic or asymptomatic. But the important point there is that they are still able to spread the virus. So coronavirus is a viral infection, and there are other coronaviruses that we know about, but this is a novel version. And symptoms, as we know, develop 2 to 14 days after an exposure. Transmission, there's been some controversy about whether or not it's airborne, but we definitely know that there's droplet transmission that is propagated through coughing, speaking, singing. There's also contact, so hand to face, surface, hand to face, and there's even fecal oral contamination. The progression of disease is over a course of 2 to 3 weeks. And most patients, we have to understand, most people will recover and have a mild to moderate course. However, there are a % that will go on to have severe disease. That severe disease is characterized by development of pneumonia or progression to something called acute respiratory distress syndrome, and may need mechanical ventilation. However, over the course of these few months and being in the hospital, we've also observed other manifestations. It's not just a respiratory disease, but it can lead to cardiac dysfunction, kidney dysfunction, and even neurologic dysfunction. And just briefly, and these slides can be shared later, acute respiratory distress syndrome is different than pneumonia. It's a complication, if you will, where fluid builds up, and then therefore those lungs cannot oxygenate your body. And those are mild to moderate course, and now you become severely short of breath, you're labored, and you have low blood pressure and potentially develop confusion. Complications of ARDS include hypercoagulability leading to blood clots, pneumothorax because of the high pressures required to sustain mechanical ventilation, and you're subjected to secondary infections. And then long-term effects we're only discovering that potentially there could be long-term sequelae of having ARDS, including pulmonary fibrosis. So let's look at Pompe disease specifically. And what I did was in real time, trying to understand how my patients could be vulnerable and looking at each of the lysosomal storage disorders. And it was actually one of my own patients that prompted me to really look into this, to start an outreach to all of my patients as the pandemic hit New York City. So when we think about the respiratory dysfunction of pneumonia and potential ARDS, you can only imagine that if you start with a baseline comorbid pulmonary disease, it could be exacerbated, or it could exacerbate your underlying disease. So we do know from the CDC and others around the world that there's high risk in patients with pulmonary disease, such as emphysema, COPD, and even mild to moderate asthma. So in our rare disease community with Pompe disease, we have to educate our colleagues, our frontline ER physicians and ICU docs, to tell them what is Pompe disease. Because as many of you know, most doctors are aware of infantile forms of Pompe, right? But they don't necessarily understand or know about the attenuated or adult onset forms of Pompe disease. And so it was critical for me to communicate to my colleagues in the front lines, just in case they saw one of my patients, to understand that this is indeed potentially a higher risk factor for them. So as we know, in our adult patients, they can have impaired diaphragmatic function or weakness, and they have need for CPAP or BiPAP at night, and they may have an impaired cough reflex. So that was very important information so that we understand how to adequately treat. In the midst of the pandemic, a lot of patients were told to remain at home until they were very severe because of the overwhelming influx to our hospital. However, with our patients with underlying neuromuscular disease, and specifically with diaphragmatic dysfunction or pulmonary dysfunction, we may need to refer them sooner to the emergency room and for hospitalization because of their underlying disease. There's also cardiac dysfunction that's been observed with COVID-19, and it's not clear. We think there might be some element of myocarditis in some patients. We did see sudden cardiac death even in young patients. And so there's also potential prolongation of the QT interval that was associated with one of the experimental drugs. So we have to understand, obviously, again, in our infantile patients, they are affected with hypertrophic cardiomyopathy, but even in our later onset patients, they may not have cardiomyopathy as we see in their infantile forms, but we do see some potential arrhythmias. And so this all had to be taken into consideration. And our liver and spleen. So typically in COVID-19, in more severe forms of it, we start to see liver dysfunction, and we see elevation in the liver enzymes, we see potential clotting factor issues, and so it's called synthetic dysfunction, and then leading to a hypercoagulable state. In Pompe disease, generally a glycogen storage disease, there is normal liver function, but they might have mild elevations in LFTs and CPK at baseline. It may not automatically indicate a worse outcome of COVID-19, but it needs to be understood by our colleagues. And as I mentioned before, as I'm a neurologist by training, and being a consultant in the wards, we did see, initially we didn't know if there was a direct neurologic effect of COVID-19 on the central nervous system and the peripheral nervous system. But sadly, we did start to see that there was an increased risk of seizures with or without related to the fever and electrolyte disturbances. But we saw increased stroke due to the increased clotting, the coagulability of the blood. We saw encephalitis. We started to see peripheral neuropathy and autonomic dysfunction, including Guillain-Barré syndrome. So if you can imagine in Pompe disease, where we have muscle weakness, and possibly already a compromised cough, and compromised ability to fight off infection, we now, if the patient develops complications of COVID and lead to a prolonged hospitalization, we could have further muscle breakdown and atrophy. Patients without a neuromuscular disease have a long rehab recovery after they're extubated. And that leads to anytime someone's hospitalized for a long time, there's potential for muscle atrophy and breakdown, especially if you're intubated. And so we're seeing our patients without rare disease requiring prolonged rehab after the acute hospitalization. And finally, renal dysfunction. We are seeing the increased need for dialysis in patients with COVID-19. Fortunately, in our patients, we typically, with Pompe, have normal kidney function. But I do want to stress that this is the minority of patients that go on to have severe complications. And it's important to understand that we don't know if there's an independent risk factor for just having a lysosomal storage disorder, but we can imagine that our patients with more severe features of a disease might be at risk of these complications. So how did COVID-19 indirectly affect our patients? Well, if you can imagine, when the pandemic started to hit, we recommended to our patients to remain at home and to avoid contact in large groups. And so what became on the minds of my patients were, how do I safely obtain my enzyme infusions? And they were scared. They were scared to enter into hospitals where there was a high COVID admission rate, and they were scared to have healthcare workers into the home. And so we instituted screening the patients and even the healthcare workers by phone before the appointments, at the beginning of the appointment. We were ensuring that all nurses were mask-gloved and using appropriate protective equipment, because now we know that there is potential for pre-symptomatic spread. And so we ensured that the staff were wearing masks, the patients were wearing masks. My staff and I called to each of our patients' infusion centers to ensure that they were spacing the infusion chairs, reducing the number of patients in a particular time slot at the infusion center to approximate that physical distancing that's really important in preventing spread. And we went as far as to see whether or not the nurses were dedicated to that unit or if they had to be redeployed. At the height of pandemic, we started to have a shortage of healthcare workers, and a lot of us were reassigned into areas that were not our normal area of specialty. And so fortunately at NYU, we had nurses that were dedicated to the infusion center, and they were not redeployed to reduce that potential exposure to our vulnerable patients. And so as this starts to hit other areas of the country, these are the things that we want our patients and our providers asking about their infusion centers. So some patients were so scared of exposure that they did defer their infusion. Ideally, we don't want to defer during the pandemic. We know that this is a progressive disorder, and as such, every treatment is important to make sure that we have continued stabilization. However, in certain cases, it was too risky. And so some of our patients did defer infusions, and we made sure we kept in contact and reassessed every couple of weeks whether or not the patient was at increasing risk. Missing a couple of infusions here and there may not make a huge impact in the short term or long term. But if, as we know, that this COVID-19 has not necessarily gone away completely, we had to think of ways to get our patients to the infusions, because this might come back in the fall and in the winter. And so with each patient, you really need to make an individual risk assessment with your medical provider and review your risk of deferring it versus the exposure, and to reassess periodically. So in general, I think most of us are aware how to prevent exposure to COVID-19. There was, initially there was competing or contradicting guidelines about mask wearing, but what we know now, especially from our healthcare workers, including myself, that even using a surgical mask or a cloth mask significantly reduces the spread of the virus to others. So it may not completely reduce your risk, but if those around you are wearing a mask, that significantly reduces the transmission of the virus to you. And so as the disease began to spread within our community, you saw that New York City residents were starting to mask. And we did indeed not only flatten the curve, we bent it all the way down. And so now we have very little infection rates going on. So we limited contact with others, we limited the congregation of size of people, tried to stay within our families, only venture out for groceries and medical appointments and exercise. And for our children, we went to homeschooling, remote learning for our college level adolescents. And then even those of my patients who were employed, most patients were eventually compelled to work from home because they were non-essential. But for my patients who were essential workers, we did do a risk assessment and suggested that they remove themselves from direct contact. So I had some nurses and doctors that I removed from initial frontline work. And I provided a letter outlining the potential risk of COVID. And that's something that you could do with your provider where you are around the country if there is a sudden surge of infections again. So in general, when outside in the community, we encourage you to wear a cloth mask, to maintain physical distancing of at least 6 feet. Don't touch your face because that's a mode of entry, and use hand sanitizer or wash simply with soap and water for 20 seconds when you return home. What happens if you're exposed? So just quickly, think about contacting not only your primary care physician, but your Pompe expert who's managing your care. You want to self-isolate immediately so that you don't expose others, and then you want to monitor your temperature and note the development of other symptoms. And then when to go to the ER. Now again, this is very individualized, because if you're very mild or pre-symptomatic or relatively mildly symptomatic with your disease, you may not have to rush to the emergency room. So I would implore you to contact your Pompe provider to talk about your specific risk. However, when we were in the midst of the pandemic, like I said before, a lot of patients were told to wait at home until they had significant shortness of breath. This is not necessarily the good enough guidance for our patients with pulmonary dysfunction. So again, please call your doctor to put in place an action plan. So for example, when my patients called me, I proactively talked to them before they were exposed. And we discussed when to go and what to do if you needed to go to the emergency room. And we came up with a to-go bag. In that to-go bag, I suggested that you have an updated list of medications, your list of medical conditions, your advanced directives, including your healthcare proxy, cell phone and chargers, list of primary contacts, and possibly even a letter from your Pompe doctor. And this is important so that they understand your rare disease. The doctors in the emergency room or in the ICU may not have come in contact with a Pompe patient before, especially our attenuated forms of it. And they may not understand how does Pompe disease manifest in adults. And so this is why we came up with this to-go bag. So here's just a general list. I've already gone through most of them of when to seek medical attention. And basically, it might be even earlier if you have significant underlying medical conditions. And just a brief slide a final slide on this is the ethical considerations. Again, I'm hoping as we learn from our colleagues, in New York and around the world that we don't come to this, but unfortunately, I'm hearing of similar concerns of reaching capacity in certain states, ICU capacity and ventilator capacity. In New York City, we came up very close to the edge of running out of mechanical ventilators. We actually came up to the edge of running out of dialysis machines. We were able to create an extraordinary number of ICU beds, but that was limited by whether or not we had a mechanical ventilator. So even if we had the ICU bed capacity, the ventilators were the limiting factor. And so there was a discussion about, and these are not just during this pandemic, but previously, what would you do in the setting of limited resources? And each hospital had some guidelines. An ethicist weighed in. And I met with our ethicist and discussed that there is these triage criteria of who gets a ventilator. And so we just talked about in general that a genetic or biochemical diagnosis should not automatically exclude a patient from mechanical ventilation. There are certain criteria that would, But again, it's looking to the specific patient in front of you and their underlying medical complications at that time, not their future. Not looking at someone who has a late onset form of Pompe who's functionally well and doing well. They should not be judged just solely based on a genetic diagnosis. And so we should risk stratify like we do for every other patient that walks in the door by their age, their comorbidities at the time, and their organ function at the time. And so that's why it was important for me to reach out and get some reassurance about that. Fortunately, we did not reach capacity for to need to triage that. Okay, so I'm going to pause there. I want to thank my team because during the pandemic, while I worked as a consultant in the hospital, they were there, including Kara, who's going to speak later today, Kara Anstead, Dr. Dinesh Lala, all reached out to my patients to make sure they were safe and to give guidance during the pandemic. And we are periodically checking in with them. And it was critical to have them there to support our patients with a variety of rare diseases through that time. So I'm going to pause there and see if we have any questions. Thank you.
Yeah, Dr. Lau, as a reminder, if you have questions, feel free to chat them in during the using the Q&A box. We do have one question that has come in, and that question is, my daughter was diagnosed with LOPD on her newborn screening. I am a frontline nurse and will be off maternity leave in 2 weeks and returning to work. Should I be concerned?
Yeah, that is a difficult question, right? Because we don't know what we do know, right? So infantile versus late onset is distinguished really by cardiomyopathy. There are late onset forms that are not so late onset, that they could be more childhood or even in early childhood. I would speak to your medical provider initially and understand what are her baseline needs. But in general, well, let's look at this in general. Children overall do well with COVID-19. We have to look at the function at that moment. So if she's not manifesting signs and symptoms, and you're saying that she's late onset, that her cardiac function is fine by definition if she's late onset, that her respiratory function is fine. It's hard to guide here, but in general, the children are doing fine. Now, if you have but a caveat is if you have respiratory involvement, say in childhood, because your form is more juvenile or childhood, then you might be at increased risk. It is a really difficult decision for all of us who are healthcare workers because we don't want to expose our family members. So even though it's a rare disease, we have to look at it as everyone else who has a potential vulnerable person in your home. And so the discussion is, what do you do? Do you simply have the children in the home, and then, there's certain guidelines we could talk about. For instance, when I come home, I make sure I shower and change and I remove all my clothing from the hospital. Because this is over the long term, you can't really isolate from your children forever. Personally, I sent them away during the height of it, and I have certain vulnerable aspects to my children. But at this point, we have to figure out how to do this over the long term. And so safe practices. We know that masks, if you are masking at work and you're potentially reducing your risk dramatically, then you can feel more reassured. You have some control over that. It's a difficult decision because, as you can see, we don't even know the best solution for school. What are we going to do with our children going back to school in the fall? And it's all on our minds, but we do know, I'm getting more reassurance about our pediatric population. But if you have underlying medical problems now, then I would put them at an increased risk factor, and you might choose to isolate differently. It's a bigger discussion maybe for another time. Hopefully that helps.
Oh, I was muted. Yeah. Well, we'll take one more question right now, and then we'll kind of move on to Dr. Law's second topic today, but she is going to have a Q&A at the end of that session as well, so we can go back to any other questions we weren't able to get to. But the one other question that came in was, I've been given the option of switching from an in-hospital infusion to one at home. This seems way riskier. What would you advise?
Oh, yeah. So I'm glad. So this has been on my mind as well. So it depends on the hospital setting and it depends on the home setting. So it really, you have to speak to your infusion agency. So in one scenario, the infusion agency were using nurses that were exclusive. So they were not redeployed to COVID patients. And but in another scenario, the infusion nurse was deployed to the ER. So obviously we tried to minimize that. But again, you have some control over the situation. It's not necessarily riskier to have them come into your home. You have the same practices, N95 on the nurse who's protecting herself and to you, hand hygiene. So it's not like she's going to walk in and you're just going to absolutely get exposed. Start the infusion and then maintain 6 feet apart. Make sure all surfaces are wiped down. That she's touched, she should be using gloves. So we've been able to effectively have home infusions in the height of the pandemic. My patients, I have not just Pompe, but I have over 160 patients on infusions. And the home infusions to date, I have not had any exposures from healthcare workers. Their exposure was from the community. I've had 26 suspected COVID patients, COVID illnesses. And of those, 6 were confirmed. None were Pompe disease. You can safely infuse. You just have to be vigilant, just like you're doing everything else. Sometimes the hospital centers are more risky if they're not cohorted. So for instance, at NYU, my infusion agency, even though it's hospital-based, is actually a separate building and it's a separate entrance. There are ways around it. Talk to your provider and talk to your case manager, because it was integral to work with pharma to help me identify each of my nursing agencies. It was the top priority for me to make sure that the infusion, whether it was in center or in home, was safe as possible, because this is going to be here for the next year or two. We have to figure out how to do this safely, and so far we are. I feel more reassured now, having come through it myself. I mean, I was exposed constantly, but I safely was masked and gowned. You can do the same. Oh, face shields. Face shields are excellent. I would definitely say face shields with a surgical mask. That's also that's important because of the potential for droplets into the eyes. Skipping, just a quick one other thing. Your individual provider should be tracking if you're off of ERT. There's a question about, could you speak to efforts to track? I don't know if there's any global or national efforts. Individually, in my own practice, I keep a list of who skipped ERT, and then we reassess periodically. Masking. Oh, the mask. I have a 5-year-old. She was 4. She was masking as well. Start practicing wearing a mask. But again, I think maybe MDA can do a different forum about this, but going back to school is a big issue for most of us in the community.
Yeah, and Dr. Law, I would like to say we are doing the end of the month of Facebook Live, all focused around going back to school. I know there's still a lot of questions and a lot of school districts trying to determine what to do. So do be on the lookout. It will be the end of July on our Muscular Dystrophy Association Facebook Live page.
Yeah, I want to highlight that this is not just a Pompe issue. It's every disease out there. If your child has a chronic medical condition, it's on your mind. And so I would say, look to the resources from the MDA and others and see how we're doing it. And the American Academy of Pediatrics has come out with a statement about school as well that you can look on their website. Okay?
Yeah. Dr. Lau, I know we've got a few more questions on here, but it might be good for us to move on to the research and clinical trials topic, and then we'll save about 5-10 minutes at the end of that presentation, and we can answer questions on the research and clinical trials presentation, and then we can go back to some of these as well if time allows.
Excellent.
Perfect. All right, take it away.
Okay, so again, it's a little bit heavy on my voice today, so hopefully you're following. And all right, so we're going to switch gears now. And I was given the topic or the idea of today, or this particular part of the conference, is to talk about clinical research in Pompe disease. It's a huge topic, and we could take it in so many different directions. But I figured I'm going to talk a little bit today. Again, here are my disclosures. I'll be talking about some experimental therapies as well as off-label use in kids. So I want to talk about some general considerations about participation in a clinical research trial. It's important for our patients to understand and parents to understand what it means to be in a clinical research trial. We'll talk a little bit about how the biochemical basis of our lysosomal disorders leads to a spectrum of disease, because that leads into our therapeutic approaches. And it's not meant to be comprehensive. I'm going to touch upon the various strategies to treat Pompe disease, but that's going to be a lead into our roundtable discussion with some wonderful experts in the field. Okay. So what are clinical trials? It seems like a very easy question to answer, but just to make sure that we're all on the same page, these are specifically research studies in humans to evaluate the effects of an intervention health outcome. Now that intervention may not be, or no intervention. It may simply to look at, you know, non-interventional, so prevention, screening, diagnosis, natural history. It could be behavioral studies. It could be quality of life. So it's not just about getting a medication and administering it and seeing if it works. There are so many different types of clinical trials out there. When you look at clinicaltrials.gov, you're looking to see what type of trial is it. But in general, all of these trials are looking at an approved protocol. This is a study outline that has to be approved by an ethics board at the institution. And then it's run by an investigator and it's approved. Then you recruit the patients to participate. And you can limit the participation of the patients. Not everyone can. You look at the data, you do a statistical analysis to see if there is any objective difference in what you're observing, and then you present it, and then it's published. And sometimes that data is then used to go on to approve a drug by the FDA. This is a very quick overview. But clinical research is much different than medical care. And so patients sometimes have a misconception that a clinical trial is definitely a means to better medical care. While we want better medical care, the purpose of a clinical trial is actually to generate more information about that disease state, and that will be helpful in the future. A potential participating in a clinical research study may not be of benefit to you in that time period. It might be beneficial in the future. And that's why we have to understand that before we enter into clinical research. What type of research are you doing? And what is the potential benefit, if there is any, versus just going along with your routine medical care. So when we look at the drug development, it's a very long path to reach FDA approval that I'm sure most of you are aware of, because our enzyme therapy for Pompe took many years to develop. And so we start off with that preclinical phase over here in the corner. And that's usually testing in the lab, and then onto an animal, either animals of just general small animals, large animals, or animals that are mimicking the disease state, so animal models of the disease, to see if we can figure out toxicity of intervention. So now we're talking about interventional trials, and to see whether or not that there's a toxicity to it, to understand dosing, to understand the mechanism. Why does the drug work? How does it work? Where does it work? What's a potential dose? But we're doing this in animals. And animals are vastly different than humans, but we learn a lot at this stage, and this stage could take years. And once they start to develop and understand this potential drug, then they apply for an IND. And that's where we start to jump over to what we call that first in human trial, and that's a phase 1 trial. Now these numbers here are for these large, for these disease states that are not rare. But in general, phase 1 trials are small numbers of patients, and they're briefly exposed to the drug, okay? And we look to see, first and foremost, whether it is safe or not, and toxicity, okay? And then from there, if it initially looks quite safe and they have a good idea of a dose, they'll move on to phase 2. And as you can see here, it depends on the study that you look at, but anywhere from 50 to 60% may move on to phase 2. So there is a huge gap between preclinical and clinical. Many trials die in preclinical and others get to phase 1, and up to 40–45% may never move on to phase 2 because they don't show either safety or because of safety issues. Phase 2 is now a little bit larger trial, and we're still looking first and foremost at safety first, and then we start to see is there any kind of efficacy. And then it'll be a larger number of patients who are exposed to the drug longer, and we're looking at over maybe now, instead of a few like months, days to weeks, we're looking at months to a year. And from there, only about a third might move on to phase 3. And phase 3 is usually a pivotal trial showing efficacy. So larger number of patients are involved, and we're seeing a longer duration, so maybe over up to a year or 2 years. And we're looking for efficacy on top of longer term safety. And from here, still about 40% don't move on to that next phase, which is the approval process, where they go to the FDA with their data and try to sit down and convince them and say, this is working. And once the FDA approves it, then there's a post-marketing phase where some companies, especially in rare disease, have to track it over the long term. Again, still doing studies on the long-term efficacy and in the real-world application of it. So just a more simplified slide here, again, showing more numbers, right? In rare disease, these are not 20 to 80 participants. It's like a couple. And phase 2 are maybe, several. Phase 3 are now tens and dozens, and phase 4 are hundreds. So the scale is different, but the idea is the same, okay? So when we look at, again, this is just a table showing that. So common versus rare diseases, showing the differences in number, but the similarities between, testing a blood pressure medication versus a medication for Pompe disease is they still have certain criteria. You have to meet the inclusion and exclusion criteria to be in a trial. So patients will feel like, well, you're developing this drug that potentially could help me. I deserve access to it. However, the company or the institutions that's developing it have strict criteria to allow certain patients in. And that's because they need to show safety and efficacy in a short time period before they start to allow others on that drug. And usually in rare disease, we have fewer number of locations and it's harder to recruit patients. So again, the idea of preclinical is, does the treatment work as hypothesized? Phase 1, does it work like it did in humans? What's the best dose in phase 2? And phase 3, using that dose, is it treatment effective and safe? So why would someone join a clinical trial? So, even if it's not an interventional, people will join because they want to advance the understanding of the disease state. So for example, what was critical to developing drugs for rare diseases are these natural history studies that we've heard about to define the spectrum of a rare disease. So for instance, when we first learned of Pompe disease, we understood the infantile form, which is this rapidly progressive disorder that leads to cardiomyopathy, hypotonia, muscle weakness, and rapid decline without therapy. However, what we understood later when we were able to start to sequence and do DNA sequencing and understanding or doing the biopsies of older onset or adult onset Pompe disease, there was a whole different type of disease out there, but it was the same genetic defect and the same enzyme deficiency, but just differed in the type of gene mutation. And so there was critical to understand how this adult onset or even the juvenile onset form of Pompe disease changes over time. Because if you can imagine, if you're trying to develop a therapy and it's targeting one type of the disease but not the other, we may not be fully treating the disease, or maybe it's not as efficacious. Other reasons to join a trial: there's no treatment available. This is from the patient perspective. It's available but not as effective. So maybe the enzyme therapy is just stabilizing and then you go on to decline further, or it doesn't fully rescue the phenotype. The treatment's available, but there's safety or quality of life concerns, frequent infusions. And the access to experimental treatment is at a reduced or no cost. But what are your concerns as a patient who's thinking about joining a trial? You're concerned because this is experimental. We don't know all the side effects of the drug. We have our best estimate of the safety is from animal studies. So a phase 1 trial is quite risky for that first in human trial to understand how does this drug affect a human? Are the side effects the same? Are there potential unknown side effects? What is the long-term side effects? Other concerns include the invasiveness of the procedures, whether it's to do the actual intervention, to administer the medication, or to monitor the medication. Does it require muscle biopsies? Does it require lumbar punctures? Does it require more procedures that require sedation? Children taking a drug untested. It's a little cut off. That's always another concern. Number of blood draws for them, perceived risk for physical harm. Maybe there is no benefit. Certain natural history studies, you undergo biopsies or lumbar punctures, and there's no specific benefit to you personally. The consent process is very important, but it might be complex. What am I signing here? What is this document saying that I'm signing to be part of this study? And then of course, what's on the mind of everyone is, what if it's a placebo-controlled trial and I'm randomized to placebo? What does that mean? How can I let that happen? And not knowing whether or not you're on active or placebo drug for a year or even 2 years. And so these are all concerns of our patients who are thinking about being in a trial. But then there's logistical concerns, your work schedule, how do I get to and from the site, compensation, how do I care for my children if I'm in the trial or if my child's in the trial, how do I get around school, the length of the study visits and the frequency. These are all in the minds of our patients. And as a principal investigator, I kind of hover between the caregiver, right, the medical doctor for my patients, and then the principal investigator. And I have to take into consideration the burden on my patient of being in a trial. And you can imagine during the COVID pandemic, how this really affected. Do I bring my patients on site? How do I protect them while still allowing and going forward with a clinical trial? It was a whole other bunch of concerns as a investigator with patients. As a medical doctor, I was trying to care for them. And then as their principal investigator of my subjects, I was trying to figure out, how do I safely conduct this during a pandemic? Okay, so clinical research versus medical care. Sometimes patients who are trial participants, they're so keen and eager to get access to a new drug that they may not adequately appreciate the purpose of the research study or the methods. They may not understand all the risks involved. And it's really important for the principal investigator to sit down with the patient and the parents to discuss what are the potential benefits, what are the potential risks and alternatives, okay? And other concerns include care costs, routine costs, and non-medical things that I mentioned. And other concerns, nobody wants to be in the control group. No one wants to be, their future is put to the whim of a randomization. I could be, 50-50, or is it 2-to-1 ratio? And blinding. How do we, conduct our lives while we're on this unknown, whether or not we're on an active drug or not? But then there's this other aspect called expanded access, where we get some patients are allowed access to experimental treatment that don't qualify for a clinical trial. Not all pharma and studies can offer this aspect, and sometimes it's offered after they've established some kind of safety and efficacy with phase 1, 2, and 3 trials. But it's usually when it's very serious or life-threatening and there's no other options out there. And again, this is not, the downside to this is that we don't know, we're not necessarily collecting information about efficacy, and we're just responding very quickly, okay? So if you participate in a trial, it's important that you understand the commitment, you understand the eligibility criteria, because not everyone who's screened is allowed to participate. If you can't meet certain criteria of walking or if it's meant for your pulmonary function, you can't be too sick or you can't be too well sometimes. You can always drop out at any time, and you have to be able to understand the language that's being used and feel comfortable asking questions of your investigator. and to understand the study goals and the outcomes. What's the timeline or the procedure? What happens after the trial is over? Do I still get access to the drug? Is there an extension study? And these are all specific information that a trial participant should be asking. And part of your role is to adhere to the protocol. And it's more strict than medical care. You have to come in at designated time points. You have to follow the protocol. And there are certain criteria that if you don't follow it, you're withdrawn from the protocol. And the biggest thing is, what if I'm on another experimental drug or a supplement that's not approved? You can't, typically we cannot test 2 drugs at the same time. We can't have you on 2 separate research drugs because we don't know which drug is working. And so you would have to make that choice. Am I on this drug or that drug? Okay. So questions to ask, what's the trial design? How long is it? What's the safety to date? Do we know anything about efficacy? What's the criteria? What do I have to do? And what are the alternatives? I might be running this trial, but as a clinical provider for lysosomal storage patients, I will absolutely tell you about the other trials out there, whether or not I'm running them, because it's important to understand what's happening in the entire field and not just what's at your hospital. Now, specifically for gene therapy, there are certain aspects that are more critical to understand. Is the dose determined? Because right now, it's not possible. We're working on it, but how to redose? What if you get the smaller dose and it's not enough? You can't really discontinue a gene therapy, right? You can discontinue going to your visits, but once you have the gene therapy infusion, it's irreversible. And you're potentially excluded from future research studies. So look at that criteria. In the current studies that I'm running, you cannot have had gene therapy before in order to be considered for another study. Okay, so what are our current therapies? Most of us already know, but to understand how do we get to where we are? We had to understand the disease first in order to tackle the problem of developing therapies. And that was done through these natural history studies, preclinical studies in animals, and studying humans. Then we used that knowledge to develop a medical intervention to at least halt the progression of this progressive disease and hopefully reverse the signs and symptoms. And so, when we think about our lysosomal storage disorders, these are enzyme therapies that were developed because their deficiency of the enzyme leads to the storage of either these sugars, sphingolipids, or in this case with Pompe disease, a form of glycogen. And that causes a disruption throughout the cell, and it's not just storage. It causes all kinds of cellular dysfunction in this complicated slide here, but inflammatory response, autophagy issues, mitochondrial dysfunction. There's a lot of dysfunction that goes beyond just having a cell being filled up with storage. And as we understand with Pompe disease, some of the enzyme activity correlates with disease severity. And so, our patients with very little enzyme activity have more of the infantile phenotype versus the other attenuated forms. I'm going to move a little fast here, but so enzyme replacement therapy was pivotal in the development of ERT was pivotal to treat a lot of our lysosomal storage disorders. It's a great, very simple concept. You're missing what community that lysosomal enzymes have this tag, this molecule, the molecular tag on it to send it to where it needs to go. And they were able to create that in a recombinant version in a petri dish and basically give it back. And so the first approved therapy was for Gaucher disease in 1991. There are some limitations, but at least this helped to reduce the storage in Gaucher disease. And so in 2006, we were able to develop, the scientific community was able to develop the recombinant enzyme therapy for Pompe disease, and it was absolutely life-saving. It was critical. It reversed the cardiomyopathy in infants. And they leveraged this fact that the naturally occurring enzyme is tagged, and it gets it to the lysosome. So understanding how the disease occurs helped us to create a therapy to target it. So here we see that, as it's traveling through the blood vessels, it gets picked up and transported to the muscle fibers by this tag. This is just to give you an understanding, because Now we're trying to figure out how to make this better. Because why? There's lots of limitations of our current ERT. It doesn't completely halt the progression, right? We understand that it was life-saving in our infants, but our infants are now have a residual phenotype left. So they're treated for their cardiomyopathy and they're living longer. And now we're seeing a different phenotype. We're seeing a partially treated enzyme deficiency. Our children still have muscle weakness. Our children still are developing, actually, we're seeing some white matter changes in the brain, things that we didn't see before because our children are surviving, the children with the infantile form. In the more attenuated form, whether it's juvenile, childhood juvenile, or adult onset, in my adults with this disease, I hear about how they're doing fine, but they still have residual weakness, or they're still progressing with those slower. And why is that? Well, the enzyme has it has poor targeting to the muscle. When it goes into your veins, as that picture showed you, it actually has to travel through the body, and it gets picked up by the liver and the spleen. And so a lot of it doesn't reach the actual target muscle, whether it's skeletal or cardiac. And muscle is a very large, quote, organ. It's throughout your body. So you have this one dose every 2 weeks, and is it really getting to where it needs to go? It has a short half-life. Even if the repeated dosing, it still doesn't approximate what your body can make every day throughout the day. And that enzyme is unstable. It's going to break down pretty quickly once it gets into your veins because it's not the environment it's supposed to be in. And so we're having difficulty penetrating certain tissues, especially if now we're seeing with infantile forms having problems with getting across the blood-brain barrier. And obviously, a lot of us know about the immunogenicity issues, hypersensitivity reactions. And in our patients, and especially with our infantile patients, the efficacy of the drug is reduced because our antibodies are attacking it. And so we have to do immune modulation before we even do that. So we need better therapies. And that's what the goal is when we think about the future of research in Pompe disease. We need improved enzyme replacement therapy, maybe fusion proteins. We're looking at substrate reduction therapies to produce less storage molecule. Chaperone therapy, that's a very interesting one where we help to guide the misfolded protein or the exogenous, the outside protein, the enzyme, and get it to the lysosome, protect it and get it there. And then we're seeing gene therapy. We do use stem cell transplantation in other lysosomes, in other rare diseases. So for instance, we have newer enzymes under investigation. We have NeoGAA, which is a modified enzyme that has potentially improved binding to get into the cell better, to better target the muscle. And that was shown in mice. And when they're running, Genzyme Sanofi is running the studies, they showed improvement in walking in their first phase 1/2, and potentially improved pulmonary function in both naive patients and switch patients. And a pivotal phase 3 trial is underway, and results hopefully will be there soon. There's a sub-study in pediatric patients. So they're taking our current enzyme and we're covering it better to get it better targeted to the muscle in a very simplified way. There's another investigation using enzyme plus the chaperone. So what is that chaperone again? It's a molecule that binds the enzyme, whether it's your own enzyme or an enzyme from outside the body, binds it, stabilizes it, and hopefully gets to that lysosome better before it gets degraded. And so there's an investigation going on by Amicus using a combination of hopefully of an enzyme that has higher tagging, that M6P tagging, and then it's administered with an oral therapy that's a chaperone. And then together, hopefully it'll bind and get to the cell better. And they did show it in mice to get to the targeted muscle, to stabilize that enzyme so it doesn't get broken down, to sustain the half-life. And it actually showed reversal of muscle damage and restored the defective autophagy in the muscle fibers in mice, right? But then in humans, their initial studies are showing that it's well tolerated, lower number of infusion-associated reactions, and in their initial studies showing some improved motor function in patients who are naive and even in switch patients up to 2 years out, and showing some improved respiratory function. So it's doing what it's supposed to be doing, and we're trying to show if it's better than our standard of care therapy. And also there's a phase 3 trial underway for this particular combination therapy of ERT plus chaperone. And there's including a pediatric study starting. Adjuvant therapy, just a quick, so not just providing the enzyme, but is there a way to build muscle? And there's clenbuterol. So again, there's lots of different studies out there, but the big thing that's on everyone's mind is gene therapy. And when we think about gene therapy, what is that? So the concept, there's 2 types of gene therapy, in vivo and ex vivo. And in vivo therapy is basically a virus that's been, I kind of simplified language, take a virus and remove all its dangerous parts as best as possible, but leave in it its ability to infect, and then put inside of it the particular gene that you need to, that's dysfunction, that's not functioning properly. So in this case, we're gonna put in the alpha glucosidase into that gene, into the capsid, the protein of the virus. And through intravenous administration in this case, and then target that somewhere. And it's going to get to the cell and then release its contents, that DNA, and that DNA will sit there and get expressed, right? Because you need that enzyme, and that enzyme is created from DNA. So the DNA is that instruction manual on how to build a protein. So instead of giving the enzyme, the protein back every 2 weeks, we're giving the instructions to the cell on how to build an appropriate one. It's kind of like teaching the person to fish. Instead of giving fish every week, you're actually teaching the cell how to create a protein that works. And hopefully that's lifelong. So it's a one-time injection. So a one and done injection, and hopefully targeting the tissue that you need. We'll talk a little bit about how there's different tissues that we're targeting here in Pompe disease. And we want to help with immune reactions. We sometimes have to suppress the immune system to give this, so that we allow the virus to attack, to kind of invade and inject its contents, and then go away after. So what we're doing in gene therapy for Pompe disease is we're trying to permeate the heart, the muscle, the skeletal muscle, the respiratory muscles, and in infantile, potentially even the CNS. And so we need to choose the appropriate virus, because each virus has a tropism or a desire to go to a particular tissue. And then you want continuous excretion of the enzyme so that you're constantly making enzyme throughout the day, because muscle is massive. You have a lot of muscle in your body, and we need to understand how much to deliver and hopefully improve our immune function to it. Our infants that are attacking the enzyme, can we improve that with consistent exposure? Do we need to do some kind of immunomodulation to make sure that we can transduce everything? So a couple of therapies out there, many companies are on the scene, and the first one, first target is liver. So why are we targeting the liver when in Pompe disease, which is it is a glycogen storage disorder, but it doesn't particularly cause liver dysfunction. But the liver is this great big organ that takes up, it could take up a lot of the viral particles and then just become a little power plant. That's the idea. It's not that we're treating the liver per se, but the liver is now going to become this little power plant or to build and secrete the enzyme, okay? And so when the enzyme is built and made, it gets secreted into the blood system, then it travels to where it needs to go. And so it goes to the muscle, to the heart, and maybe to the brain. And sometimes depending on the company, they may not only just transduce it with a viral vector that goes to the liver, but the gene might have some changes on it to make it more enhanced to express itself. So it's a little complicated, but maybe it would be even more enhanced to express. And so we see Spark Therapeutics involved, we see Actus 101 being developed, Sarepta and Abuena. So there's several, there's a in vivo liver targeted gene therapies that are in research studies right now, including in humans. Muscle targeted, I want to comment. So the muscle is the problem in Pompe disease. So why not just target the muscle? Well, the muscle is so vast in the body that we had to figure out how, can you really give a high dose of virus? Is that safe? And so what they're developing in Audentes is a AAV8 to kind of targeting a variety of tissues, including cardiac and skeletal muscle, liver, and spinal cord. And they show that in the mouse model. And the phase 1/2 is opening in humans now. And so they're hoping that this particular AAV8 serotype will get to the skeletal muscle directly. So this is different than the liver-directed one. And the last word on ex vivo is a little bit complicated. It's basically you're taking the patient's cells out of the body and you're infecting those cells in the Petri dish, and you're giving those cells whatever gene you want, modified in whatever way to make it more enhanced. Then you give it back to the human, but you have to give them immune modulation, a modified bone marrow transplantation regimen. And so you have to immunosuppress the patient, and then those cells will go in and need to engraft into the bone marrow, and eventually it becomes, or it differentiates into the various secretes the enzyme. So AvroBIO is big in ex vivo gene therapy. They've developed it for other lysosomal storage disorders, and they're in preclinical development for Pompe disease. But what are these challenges? So again, this is a very quick overview, but there's challenges in this. It's not a cure. We still have abnormal protein being made. Is that still problematic? Still doesn't reverse end organ damage, right? So if you already have damage, you can't reverse that damage. You'll halt the progression of wherever you're at and hopefully stabilize. And we still need to understand the post-treatment phenotype. What is the residual disease that's left that's not being treated by this? Just like we saw with enzyme replacement therapy and the white matter disease that may develop in infants with Pompe disease. Are we able to target all the organs? Do we need to do multiple organs like they are doing in Audentes? And what type of viral vector to use? We still have to worry about antibodies, this time to the viral vector. A lot of the criteria for inclusion in a study right now for gene therapy is you can't have had any kind of antibodies to that virus by accidental exposure over time. You're excluded from that. So in certain studies that I've run, up to 30% of patients were not eligible because they have antibodies to the virus, because that's just going to completely neutralize it. Will it last? We don't know yet, right? This is just new. Will the expression of the gene last? And right now we can't redose. It's not safe enough to redose. There are efforts, and maybe our moderator, our panelists can talk to that later about the potential for redosing. A lot of gene therapies start with a low dose, a medium dose, and high dose. And those first few patients that are in that first cohort that are so critical to our understanding of the safety of this may not benefit because it's too low of a dose. But we can't necessarily go straight out with a high dose because of safety issues. And then these patients are no longer eligible for future research programs in a lot of cases. Maybe that'll change. But right now, a lot of the protocols are saying you cannot have been in another trial with gene therapy, okay? So gene therapy may not only be the only therapy. We still need, right, a multidisciplinary approach to our patients. We still need to have muscle conditioning and prevent atrophy. Maybe there's still a role for small molecules and chaperones. It may not be enough. We can't just simply give the gene back and let it go. We have to condition the body. And then this is just the last slide, is just to understand that once we start to rescue or give that enzyme back, we need to understand the residual disease there. So in general, drug development for rare diseases is a long process and involves multiple phases. Pompe disease is a specific enzyme deficiency that leads to multisystemic manifestations and age of the patient. And that dysfunction arises not just from storage, but all other disturbances in the cell. And so we have to think about therapeutic approaches that not only correct that enzyme deficiency, but correct those other cellular dysfunction, whether it's inflammatory aspects or mitochondrial or autophagy. Early intervention is key, and that will lead into early diagnosis, which is often difficult in our rare disease population. And maybe there isn't just one approach. Maybe it's a combination approach to therapy. And so we need to understand that when we're thinking about new therapies. Okay, thank you. So I'm going to pause there.
Wonderful. Thank you so much, Dr. Lau. That was fantastic. There was a lot of wonderful information shared there. We are kind of at 12:10, but I do want to offer a little bit of maybe 5 minutes for questions. So if there are any questions related to the research and clinical trials topic or the COVID-19 topic that Dr. Lau had discussed, please feel free to chat those into the Q&A box. And we will we'll go ahead and take just a few minutes and answer questions within there. Dr. Lau, we do have one question that was still in the Q&A box from the COVID side, and it was really around a child who goes back and forth between two households, and that the families are really quarantining in different ways. And the question really is, how does this child affect the family that has a child with Pompe, especially if the child keeps going back and forth and they're traveling out of state?
Right. So that's you know, we can take it away from even just Pompe, just in general, a vulnerable child, and then someone's coming in and out of the home. The same precautions need to be applied. You have to feel that you can trust that the other family is adhering to guidelines of reducing exposure. There's only so much you can do. Obviously, the travel itself is important, just like we're thinking about travel to and from an infusion center or anywhere else. You know, whether it's a private car back and forth, that's safer than mass transit or flying. It's still not clear that flying, how well we can reduce exposure. So you have to look at each of the aspects of the exposure. Is the child in the other family, is that family adhering to social distancing and masking guidelines and hand washing? And is that travel to and from? It's not just being in and out of state, right? I was from New York State, and people would, oh, you're from New York. But if you're actually, you know, traveling by a solo car, that's different than, say, putting the person on a train with other members, with other people. So you really need to look at the risk at each level. It's not just a clear-cut answer of like, absolutely not. You know, here in New York, we're trying to expand our bubbles a little bit. How can we be so isolated from all our family members? And so you have to really talk with that other family and talk about, how do we risk reduce? Do we expand our bubble to be 2 families? And how do we expand it safely? And that's, I think a lot of us are trying to deal with that right now. And so it is a risk, but it's not an absolute no or yes. You have to talk to that other family and have open lines of communication on what risky activities. Okay. The Washington Infusion Center closed. Well, right, I would say, you know, you're not absolutely, can you wash infusion center clothes with regular laundry? In general, probably fine. High heat, if you're really concerned about that, you could do a separate load. You're not automatically carrying the virus on you. It's about exposure. For me, as a healthcare worker, I was, you know, obviously exposed in a room with COVID positive patients. I would separately wash my clothes. You're just simply it's kind of like out and about in general. So it's not specifically COVID positive clothing. So you could wash it with regular laundry on high heat. If a child starts ERT at 9 months, how long can we expect the treatment to be effective for? Will there come a time when ERT won't work? That's a complicated question, because I'm not sure if the child has infantile. It sounds like infantile. I'm not sure if that was, for those of you who are not familiar with infantile forms, because you have an attenuated form, the infantile forms, they don't actually make a lot of protein, so they attack. Their body thinks the enzyme is foreign, and it will attack it, and it will actually neutralize and destroy the enzyme as it's going in. So as long as the neutralizing antibodies don't exist, or they're low enough, it should work. You're clearing out storage. The question is, did you have enough muscle development in utero and perinatal period? Are you going to be able to sustain it? And that's what we're not sure. So is it working? As long as it's clearing storage, it is working. But you're absolutely right. Do we need other adjuvant therapies to promote muscle growth and other aspects of it? So that's why it's not the only answer. ERT is not the only treatment. We have to look at it holistically. Okay, you've mentioned enzyme activity corresponds with severity. My daughter was diagnosed via genetic testing. Her enzyme levels were never tested. Would you recommend enzyme testing even after diagnosis? Another complicated question, but I'll try to answer it in this time frame. So in general, I always say, don't look at the exact amount of your enzyme to say, this is how bad I am. No, it's more of the big groupings between infantile and non-infantile. And so as a, say we get a new genetic variation, we've never seen this mutation before, we can kind of predict a little bit based on the enzyme activity, but it really, you have to look to your patient, to your daughter. Does your daughter have cardiomyopathy? Then she has the severe form, right? Severe, quote, severe. Does she not have cardiomyopathy? Then she has the attenuated form. But I have patients that don't manifest until 30, and then I have others that manifest at 10. So that attenuated group, it's really hard to say, well, your enzyme activity is this, and therefore you're going to have onset in childhood versus adult. Do not look at that as specific. Would I recommend enzyme activity? I'd like to get it. I'm a purist. Whenever you have a genetic test, and maybe it's known mutations and you know that it's definitely pathogenic, but I do get one-time enzyme level just to understand what it is, and because people can have variations in their genes, and especially if they're not manifesting yet, and it's a new mutation or new variation, you want to look to see that the enzyme activity is affected. But it's not necessarily going to affect your prediction of severity, okay? Does that make sense? You really need to look clinically at what your daughter has. Again, severity is very variable in the attenuated forms. Okay, other questions?
I think there's one just around, I think this is the last question, and then we're going to take a small break and get a line for the drug development roundtable. There's one question about someone that has 4 siblings that were all diagnosed within a 6-month period. He is asking, he's saying, I thought it would be a good idea to do a gene study, being that 4 siblings from the same parents with 100% presentation. Since then, he states 2 of his brothers have passed away.
My brother, 2 years younger, has passed. I see it in May.
Yeah.
Why would it not be a good idea to research our genes and maybe to find out why someone presents rather than not ever presenting? So one thing there, I just want to say that everyone, with each pregnancy, you have the same risk factor. So it is odd, it may feel odd that all 4 of you had it, but it depends on the genotype of your parents. So some families may not have, they might have 1 affected, because each pregnancy you have your independent chance. So if your parents were carriers, each of you had a 75% chance of not having the disease. And by bad luck, it's the 25%, it happened each time. Now, it's complicated to understand why there is a difference in severity within families. Sometimes it is other comorbidities or other medical problems and healthcare or health lifestyle, and some things are out of our control. So as I say, tell everyone with a rare disease, the last thing you need is to have other medical problems, like control your blood pressure, control your weight, make sure that you don't smoke and alcohol and all of that, all of these other lifestyle changes. But at the end of the day, there's still a difference among siblings sometimes. And that's because we're not genetically identical. We're not genetically identical. And even in our identical twins, there could be differences. And so you're right, it does. If there's a researcher out there, they would want to maybe look at the differences in the genes, because there's modifier genes. Again, this is outside the scope of this talk today, but what makes us different from our siblings? There's probably other genes out there that are either helping or hurting us in different ways. So it's hard to understand exactly why your sibling passed away younger. Right. It is of interest in certain researchers to look at a family that had variable expression. I had another glycogen storage disorder that manifested as a really severe case in the child and a very mild case in the adult. And that was extremely interesting because it hadn't been reported before. So it's important. You can look on clinicaltrials.gov to see if there are researchers interested in the genetics of, or the genetics of Pompe disease. Okay.
Perfect. All right. Well, thank you so much, Dr. Lau. That was fantastic. And thank you for answering all of the questions as well that came in. We are going to take just a quick, maybe 5-minute break to get our panel all set up on screen. I will ask Dr. Lau, if you could stop sharing your screen, and Nicole will take it over. So we'll take a quick, just 5-minute break. We'll come back around 12:25 and start off with our drug development roundtable. I will say during the drug development roundtable, and it marries nicely to what Dr. Lau had just kind of shared in her topic, we do want to hear from those of you in the audience today around what your questions are, what your comments are, what your thoughts are on the drug development landscape for Pompe. We're going to have a nice, robust conversation. We've got some members from a few pharmaceutical industries with us. We have Dr. Lau and Dr. Byrne from the clinician side, and we have a family member as well. So we'll take a short 5-minute break, and we'll be back here around 12:25.